Hello and welcome to our D12 protein synthesis review video. So this one is all about how do we make proteins. There's kind of a lot going on in this one, and so we're going to go through it here. The first standard 1, is transcription. And so transcription is the process of synthesizing RNA from DNA. And so in transcription, you can think about it like transcribing. So you're taking a sequence and turning it into another nucleic acid. So it's not as different as translating, right? But it is still switching it a little bit. And so how does this happen? It's done by RNA polymerase. So in the previous DNA replication section, we learned that DNA polymerase makes it. Now we have RNA polymerase. And so this one is interesting because RNA polymerase does everything. It can do it all solo style. And so what it does is it will unwind and break the hydrogen bonds between the DNA strands. So it separates the DNA strands. So it does everything that helicase does, and it creates the new RNA strand. So it makes the polymer. It actually makes the polymer of RNA using the template. Using DNA template. And that leads us nicely into standard 2. And standard 2 says, how does it do it? It uses complementary base pairing, just like it did in DNA replication. Except now, if there's an adenine in the DNA, it will pair with uracil. Thymine still pairs with adenine, though. So this is DNA on this side, RNA on this side. The cytosine to guanine is the same. This allows us to get the instructions from the DNA, right? So this allows you to do it over and over again without any changes being made. The nice thing about the hydrogen bonding is nice because it's not a true bond, and so it can be broken relatively easily. And so the hydrogen bonding between DNA and RNA tells you you've done it correctly, and then also allows it to separate later, because you don't want to leave the RNA there, right? And so that allows that to happen. All right. What else we need to know is that it does not affect the DNA. So this does not affect the DNA. So you bond the RNA to it, and then it releases, and it does not affect the DNA. The DNA template is stable. And so you can do this over and over again without changing the DNA, which allows you to make many, many copies of the mRNA without worrying about the effects on your DNA template. And so sequences are conserved, and there's no mutations happening due to this process. One of the things that is mentioned in the standards that we want to talk about briefly is the fact that there are certain somatic or body cells that never divide. And since they never divide, they must have a perfectly conserved DNA, because they can't make any new DNA either, right? And so they have to be very careful with their DNA. They don't want any damage to happen with it. So by making this RNA, they allow the DNA to stay in the nucleus protected and not be exposed to anything outside the nuclear membrane. Right. The last part of transcription for the SL standards is gene expression. And so it's the expression of genes. So when we say we're taking RNA from DNA, it's specifically genes. And so a gene codes for a protein. Now, not every protein is active in every cell. So we have what's called the proteome, and the proteome is the proteins expressed in a cell. And it's different for different cell types. And so you can imagine, right, your neurons would have different proteins expressed than like a skin cell. And so this happens because you can turn gene expression on and off. So genes can be turned on or off. We learned about this, right, when we learned about stem cells. Stem cells they have no patterns of differentiation. Once a cell has been differentiated, that really means you turn certain genes on and off, and transcription is where that starts. You wouldn't want to, so you typically turn it off before transcription can happen. Alright, so now, for continuing on with the SL standards, we're going to move on to translation. So translation is when we actually make the protein. In this case, it's known as the synthesis of polypeptides, the precursor to a protein, from mRNA. So you're taking that mRNA sequence and you're going to use it to make a polypeptide chain, which is a chain of amino acids which can fold into a protein. Now, how does this happen? That's standard 5. So how does it happen in standard 6 and 7? But we'll start with 6 first. So you have your mRNA strand. You have your mRNA. The first thing that happens is it binds to the subunit of the small ribosome. And then the large subunit of the ribosome will appear from the top, and this is where tRNAs bind. So this is the large subunit of the ribosome, and this is where tRNA goes. Then the mRNA will, right here, will bind to the small subunit. So small is for this one, large is for the tRNAs. So the tRNAs, they look roughly like this. This is a very oversimplified drawing, but they look like this. They have the amino acid at the top. Then they have this part down here called the anticodon. And then every 3 bases of mRNA is called a codon. So anticodons and codons will go together. That is the next standard. That's standard 7. But basically, the ribosome, for the SL level, the ribosome is responsible for the polypeptide part. It is responsible for taking the amino acids and covalently bonding them. And that covalent bond has a special name because we're biologists, called a peptide bond. It's really just a covalent bond between amino acids. So in the ribosome, you'd have this happening. You'd be bonding together all of the amino acids like this with the covalent bonds, and then they'd be held on like a tRNA, and then a new tRNA would come in, and you're going to transfer it to the next one over and over. So that's the purpose of the large subunit. The small subunit is moving down the mRNA and holding the mRNA in place. So the small unit holds the mRNA in place. Now we mentioned this already, but if this is our mRNA, it looks like this. Every 3 is a codon. and it will match up with the anticodon of a tRNA like this. And so you can only have 2 at a time in the ribosome, but they would match like this. And so if this one was AUG, then this one would be UAC for this anticodon here. They use the complementary base pairing rules. So they use the complementary base pairing rules, again, same as before. Alright, so that's pretty much translation for the SL level. The genetic code has 2 main things that it has. It's all about the triplet. So there's sets of 3. It's in sets of 3. Sets of 3 bases code for 1 amino acid. Now, I don't know if I can do this without the mRNA table, but so standard 9 says there's an mRNA table. And so for example, AUG codes for methionine. And it always codes for methionine in every living thing. This is called universality. Universality. And so basically what it means is, if you have the same triplet, it will be the same amino acid in plants, animals, bacteria, fungi, etc. And it allows for GMOs, or genetically modified organisms, because we can take a gene and put it in a different organism and it will still make the same protein because of the universality of the genetic code. Now, I do need an mRNA table for this next part. I'll pull one up. Now, for the next term is the term degeneracy. And what that says is all of these are proline. All 4 of these bases are proline. Or sorry, all 4 of these triplets are proline. And so this 3rd base here can change. This is called degeneracy. And what it allows for is for mutations to have less effect. So it lowers the effect of mutations. The 3rd base is redundant. The 3rd base is redundant in a lot of the amino acids. So there's 64 combinations, but only 20 amino acids. So there's 64 triplets, but only 20 amino acids. So there's redundancy, and this is called degeneracy, and it allows for you to have less effect from mutations. Okay. Oh, and I said we were done with translation, but we're not, because of course it's out of order. Standard 10 is also about translation. So if we come back up to this picture here, standard 10 says it moves stepwise along. And so really what's happening is this will move and it'll step down, and it's going to go codon by codon. And so what this means is, as it's stepping, it is continuing to increase the strand. So it's stepping and it's elongating the strand. So this leads to elongation. And so the ribosome kind of like walks along it, growing the amino acids by peptide bonding. And so it's like this here, this would be the stepwise. And so it'd move over to this spot, then this one would add it. And then it would move over to the next one and a new tRNA would come in. And then it would add this one. And so it's like walking along, adding them as it goes. Right, the last SL standard is mutation. And so mutations are ones that change protein structure. And so point mutations occur at a single base in the DNA code. And so if you have like AUG in the mRNA, AUG, CCC, CC, CC, AC. And if you change like this letter, or if you were to change this letter, especially one of the first or second letters. If you change that, you change the amino acid that it codes for. And if you change the amino acid to one that is a different type of amino acid, remember we learned there's types of amino acids, so if you change it to a different type, so like no longer a polar or a nonpolar, that could affect the structure. If you change the base that codes for cysteine, you can affect the structure. Or you can have an insertion or a deletion. These will dramatically change the structure because they shift the sequence either forward or backwards. So this would shift it like this would shift it this way, this would shift it this way. And when you shift the whole sequence like that by inserting or deleting a base, you change the protein structure. And so you can no longer have a consistent structure. All right, so that is the end of SL. For HL, we have a couple of things that we need to add in. First is the direction that we do it. So transcription and translation are a little tricky because for the first time, the direction is a little different. This transcription reads 3' to 5' and builds 5 to 3, just like in DNA replication. Translation reads 5' to 3', and it builds in amino acid chains. So it doesn't build in a direction. So it builds amino acids, which do not have the primes like our other ones. And so this one now, notice, it is reading 5' to 3' instead of the other way around. So this one is building 5' to 3', and then you're going to take that and feed it in from the direction you built it. So whatever was the start here is where you start for translation. It's the opposite of what we normally think of. That's standard 12. Standard 13, where do we start transcription? We start transcription at a region called the promoter. So you have your gene, and then you have a promoter region, and RNA polymerase will bind here to start. So RNA polymerase will bind at this region to start. And how does it know to bind at that region? There can be little things called transcription factors. And these transcription factors are often called enhancers, and they can enhance transcription. They do exactly what it sounds like. They activate transcription. And so they can bind to the promoter region. So these transcription factors will bind to the promoter, and they basically call over RNA polymerase. So they attract RNA polymerase to start transcription. And so that's how we start it. Now, I'm going to jump to 15 and then come back to 14. Once we have our mRNA, which we built 5' to 3', we're not actually done in eukaryotes. So our cells with a nucleus, we're not actually done. We do some post-transcriptional modifications. Step 1, you add a modified guanine cap. So you add a guanine cap to the 5' end. So the G stands for guanine. And you add an A tail or an adenine tail to the end. These are to protect it from degradation. So they protect from degradation. Protect from degradation. And so they stop any damage to your mRNA in the middle here by adding this cap and the poly A tail. The other thing you do is you take it and you actually chop it up. And so you have these sequences. You have introns and exons. Introns are not going to be part of the mature mRNA. Only the exons will. And so it looked like this to form the mature mRNA. And then you would add the guanine cap and the A tail. And so the way to remember this is exons will exit the nucleus to go to translation, whereas the introns will stay in the nucleus, and they actually have they work for gene expression, so they can actually like turn genes on and off. So introns do have a purpose, but their purpose is not for gene coding. And so that is the last part of Standard 15. Standard 14 is understanding that there are different sequences in DNA. So we have our genes, those are the normal ones, but then we also have regulators of gene expression, like our promoter regions. So these are other parts of DNA. DNA, not gene. We have introns, we talked about those already. They can be used for gene expression regulation too. Telomeres, this is another one. Telomeres protect the end of the DNA from damage, or the ends of your chromosomes. So there, if your chromosome looks like this DNA at the end would be telomere DNA. You also have non-protein coding genes, and those would be genes that are for RNA, such as rRNA and tRNA. You actually have over almost 200 types of RNA, but these are the main ones. rRNA makes ribosomes, and tRNA makes tRNAs. So these are sequences of DNA that code for these. So these are also found in the DNA coding. All right. Now, this last one here is an interesting one, and it's splicing of those exons. So if you have 3 exons, E1, E2, and E3, there is some evidence, not a lot, some evidence that you can change the order of these, which gives you different proteins, right? So if this was mRNA 1 and this was a different mRNA, you can see how these exons have been changed to do a different one. So this allows for one gene to code for multiple polypeptides. This is pretty rare. I was talking to a researcher this summer who said it does not happen very often, but it can happen occasionally. And so this is called splicing, or alternative splicing. All right, making alternative forms of a gene. All right, 3 standards to go. 17. Translation. How do we actually start it? Initiation. So it all starts with the small ribosome attaching to mRNA. That starts it. That's the very first part. From there, it will move and find the start codon. It finds the start codon, which is AUG, finds the start codon, and then tRNA attaches. 2 of them do. So the first one is called the initiator, and then the second one is just the next one in the sequence. So you end up with 2 tRNAs attached. So it looks something like this. Where the first one is the initiator. Oops. And so you've got, that's the first part, and then that will attract the large subunit. So then the large subunit will come in. These 2, the initiator tRNA attracts the large subunit. Now the large subunit has 3 binding sites that look like this, and they are ape in red backwards, because that's the direction you're going to go. So if this is the 5' end of the mRNA and this is the 3' end, it's going to move in this direction. You have the A site for arrival, the P site is for peptide bonding, where it's going to transfer it to this one, so it's going to transfer to that one, and then the E is the exit site. So So that's the roles of the 3 parts of the ribosome. Okay. Standard 18. Polypeptides can be modified after translation. And so basically you would have a sequence like this and you could chop off part of it. The most common one of this is pre-pro-insulin, which actually is chopped like twice, and then a part of that is actually used for something else. And so pre-pro-insulin is what gives rise to insulin, and it's actually edited twice. So you chop it up 2 different times. So you go chop off the pre part, and then you get pro-insulin, then you chop it up again and you get insulin. And so this allows you to modify it so you have your polypeptide will then become a protein. So that's why we call it a polypeptide, is because it hasn't been modified yet or folded to form a protein. The last standard just says that you have to recycle amino acids. Otherwise, you wouldn't be able to form all your proteins. And so this happens by proteasomes. So these are enzymes that degrade proteins, and they break down old ones to give you back the amino acids. So they take a protein and they turn it back into amino acids. That way you can do translation again. So you can't do protein synthesis if you're not constantly breaking down your proteins. And that gives you your proteome, which is all your functional proteins.